All right, I don't see the recording thing yet, though.
We are on. I'm sorry you didn't get the notice, but you are good to go.
All right. Welcome, everyone, back to our Musalam Nutritional Education Center. Back with some more new microbiome information, presentations, recipes. I'm glad to see everyone here. And we're going to get started. And so for those who haven't been here before, me and Katrina will be presenting together. We'll alternate every month. Next month, Sanaz will be back with us. But the whole idea is that we're going to teach you a little bit more about the microbiome and the ingredients and the foods that you could potentially be eating to help. And so Katrina's going to take the first half, and then I'll follow it up with a little bit of a cooking demonstration. If you have any questions, use the chat function. And then if you have any other questions towards the end, feel free to let us know and we will talk through everything with you. So I'm going to hand it over and I'll see you in a little bit.
Great, thank you very much. So I'm Katrina Whiteson, and I'm also here in the teaching kitchen with Chef Jess, but I'm a professor in the biochemistry department here on UCI's campus. And today we're going to have a launch of our year of culinary health lectures. And we're going to start from Microbiome 101 and healthy aging. And I'm here in the kitchen so I can see a sneak peek at the foods that Chef Jess is preparing, and you're going to get to learn how to make some great recipes. I'll post them here in a minute too. So to start from the beginning, I'm going to define microbiomes and help us think big picture about how understanding the microbiome could improve our health in the decade or so to come. And so the kinds of big picture improvements that microbiome science could help us achieve include, for example, improving cancer treatment outcomes through carefully defined dietary interventions. And so we have evidence, solid evidence, showing that people who eat more fiber have sometimes double the progress-free survival of their cancer treatment. Specifically, that was for melanoma patients on immunotherapy. So we want to know if we add fiber to people's diets as they're undergoing cancer treatment, will that help their outcomes? And so that we're doing studies on that front here at UCI right now, actually. We also are interested to know, for example, if we could treat cardiovascular disease by increasing dietary fiber rather than having to use pharmaceuticals like statins. Statins are some of the most prescribed drugs in the world, and we might be able to avoid some of the use of pharmaceuticals through the use of fiber in our diets. So overall, big picture, we could potentially reduce our dependence on pharmaceuticals or improve their efficacy with improvements in diet that are focused on helping the microbiome. Okay, so what do I mean by microbiome? Well, microbiomes are collections of microbes, bacteria, viruses, fungi, other microbes that are organisms too small to see with the naked eye in a particular environment. So we have specific microbiomes living in our gut, on our skin, in our oral cavity. Microbes are very much attracted or able to survive in environments that are able to support their particular metabolisms. There's enormous diversity of microbes. It's amazing. And so we have actually quite specific types of microbes living in different environments. So early in our lives, our bodies and especially our guts are colonized by microbes, and we're exposed to them in our household, in our environment. Our microbiomes are very much shared in our households. And so early life microbiomes are fascinating. And actually, so are microbiomes throughout our lifespans. And these messages about increasing dietary fiber become extremely important later in life for healthy aging. And so we're going to talk about that too, and how eating fiber and promoting a healthy microbiome can have really big impact on our brain health, for example. So in the last 10 or 15 years, this field has exploded, and we now have a lot of knowledge of how the composition of the different microbes are correlated with health states like obesity, cancer, autoimmune disease, brain chemistry, even whether our vaccines are effective, are all very much correlated with the kinds of microbes we have in our guts and on our bodies. And so we're in this great moment where we've got a lot of knowledge, And we're starting to design studies that are more intervention, where we can try to improve things and see how we can help improve health in all these different areas. So Chef Jess and I have a year of lectures planned again this year. It's the second year we've been doing this, and it's been really fun. I've put my email address here. I welcome questions, and this is a link to the recipes, and I'll show them to you here too. Here's the year of lectures we have planned. And so in each case, we have recipes where we're going to talk about the molecules in the food and how those impact the microbiome and health. And we have an annual series here where we have different health conditions in mind as we talk through the recipes. So here's today's recipes. Chef Jess is going to prepare a spicy beet salad. And that's a good one for getting polyphenols, which are the molecules that often make your food colorful. And they also are very good nutrients for supporting a healthy microbiome. So they definitely fit into the category of eating the rainbow, for example, and getting a lot of those healthy molecules for your microbiome going. So we've got the beet salad, we've got multi-seed crackers, and then also dark chocolate berry bark. So Chef Jess will show how these recipes work around half an hour into our lecture, but the link is there in case you want to be able to follow along. So traditionally, humans and the environment have been studied separately in terms of microbial characterization. But the microbes actually don't distinguish. And it turns out that a lot of the same approaches that environmental microbiologists have been using to study soil and oceans have turned out to be really important for studying human microbes too. So this last 10 or 20 years have been really interesting because for a long time, clinical microbiology was more focused on one pathogen at a time and not really considering all of the other microbes that live in the human body. We didn't understand how important they were to our health until more recently. Whereas the microbial ecologists studying the soil and the oceans were off developing wonderful approaches for studying the whole community of microbes together. And actually here at UCI, we have really great experts who study soil and ocean microbes in the context of climate change, for example. And so I personally have really benefited as a microbiome scientist studying humans from the expertise of the environmental microbiologists in our ecology department. And so a human body is just fully exposed to microbes in all different contexts, and we have very complex, rich communities of microbes living on our bodies. And as I mentioned, the composition of the microbes correlates with obesity, cancer, autoimmune disease, brain chemistry, and a number of other conditions. So just to start from some key postulates of microbial ecology, microbes are found in every Earth environment. And this might sound extreme, but basically nothing is sterile. It's pretty hard to find a sterile environment. For example, there's a whole program up at JPL near Caltech where they're preparing, spaceships and rockets to go out into space. And we want to make sure we don't contaminate the world, the universe with the Earth's microbes. And so we work really hard to keep those instruments sterile. But it's hard. The microbes will even eat the cleaning reagents that the people are using to try to sterilize the surface of those rockets, for example. So microbes are really clever when it comes to making use of nutrients and finding places to live, whether it's the bottom of the ocean or in a lot of medical textbooks, they'll mention that the eyes or the lungs are sterile. But actually they really aren't. It's maybe very low abundance. Urine, for example, was thought to be sterile for a long time. And we now know that there's definitely microbes in our urine, especially when you have a urinary tract infection, of course. But most microbes are not pathogens, so this is not a problem. I mean, microbes are everywhere, and it's always been that way. Humans evolved in a world that was full of microbes. Every organism did. In fact, biology and life all started there. So it's nothing to be concerned about. It's just interesting because all the microbes we hear about in the news are pathogens, but most microbes are not pathogens, and they're not looking to cause disease. They're just looking to live their lives, And then another key postulate is that every molecule can be used by a microbe, just like the cleaning reagents I mentioned that microbes can eat when we're trying to sterilize spacecraft. Microbes will find a way to use every molecule. That's a bit of a challenge. Like I would love to hear ideas of things you think a microbe couldn't make use of. Maybe glass is an idea. Microplastics are an interesting one, although there's a lot of research into how we could use microbes to break down microplastics. Anyway, so those are some of the key postulates of microbial ecology. From the perspective of the human microbiome, most of the genetic differences between any two people are encoded in the microbial communities. So any two people have very similar human genomes, but the microbes we carry have incredible genetic diversity. So for example, identical twins, they'll have identical human genomes, but especially in their guts, they'll carry a huge diversity of different types of microbes that have very complex genomes that are very different from each other. So there's a lot of genetic information in our microbial communities living on our bodies. And some of those communities are the densest on the planet. So I have this plot here showing you very low density microbial communities at the bottom, like in our stomachs, all the way up to dental plaque and colon samples, which are extremely rich with microbes, even more so than soil, for example, which is famous for being very rich with microbes. For every microbe, for every bacteria, we probably have about 10 times as many viruses infecting that bacteria. And we are now understanding in the last 15 years or so that disease can arise from having a disturbed microbial community. So from not having an ideal microbial community as our immune systems are developing earlier in life and throughout our lives. Figuring out what a healthy microbiome is a very difficult quest. It's almost becoming a philosophical question to answer, like what is healthy? But that's something our field has been tackling for quite some time, actually. So human microbial communities, as I've just mentioned, have incredible metabolic diversity. In a human, you might have easily 10,000 bacteria colonizing. And while the human genome might contain 23,000 or so genes, the microbial genes, each one carries many genes, and there's a lot of them. So you might have, say, 8 million on average different microbial genes being carried in a person. If I could collect a small amount of blood from anyone in this audience, I could probably detect about 500,000 molecules, and about half of them are produced or modified by microbes. So in a way, the microbes are impacting every corner of our body in a molecular sense because they have such complex metabolisms. And each of us is quite unique, as I've mentioned. Part of that comes from the fact that for every bacteria, there are quite a few viruses infecting the cells. And so there's interesting diversity arising because of the evolutionary arms races going on between the bacteria and the viruses. So there's a lot of new mutations arising in all of our bodies and on our guts, in our guts through the years. This is a really interesting way to look at the 20th century. And this is actually the information that first really convinced the National Institutes of Health to fund microbiome science. And if you look at the second half of the 20th century, you'll see we had this remarkable decrease in infectious disease. This also tracked with big increases in human lifespan. So these were enormous triumphs of humanity because of vaccination, sanitation, antibiotics. We had a number of public health triumphs that really helped us conquer a lot of infectious diseases. But during that same time period, we also saw big increases in autoimmune diseases and allergies. Maybe cancer and even Alzheimer's disease could belong on this list. And these increases have continued. So our health is really being affected by this, and it's a bit of a mystery. But one perspective on it is that these increases in autoimmune disease and allergies are related to a decrease in exposure to microbes. And so if you're interested in this, I can really recommend Marty Blazer's book, Missing Microbes, in which he describes, for example, how an average American child receives 18 courses of antibiotics by their 18th birthday. So we're really changing how we're exposed to microbes compared to our ancestors. And this is changing how our immune systems develop and changing our risk for these diseases, which we still don't understand the triggers for all of these very now becoming quite common autoimmune diseases that have really big impact on people's lives. And on this front, there's these really fascinating efforts to collect the microbes of human history while we still can. So for example, the Global Microbiome Conservancy is working towards collecting microbial samples from people in all different parts of the world so that we get a snapshot of what human microbiomes are like. Because the world is changing so fast, if we don't do this now, we might really never know what the microbes of humanity looked like in our past. So there's a seed vault, a microbial vault being built akin to the seed vault that you may have heard of, like a Noah's Ark for microbes. And I think it's a really fascinating whole effort. So just to give us a little bit of a tour of the human body in terms of our microbes, I don't only want to talk about the gut. That is what people probably mean when they say microbiome is gut microbiome. But for example, the saliva microbiome is a really interesting community. We have huge numbers of microbes in our oral cavity. And most of them are streptococcus. And when I first learned that most of the microbes that live in our mouth are anaerobes, which means they can't tolerate any exposure to oxygen, I found that really shocking. I mean, clearly there's oxygen in the air. So how can microbes in our mouth survive if they can't survive exposure to oxygen? But they form really fascinating gradients where the microbes on the surface will suck down all the oxygen, so the ones underneath are protected. And you can see those gradients in this image here with streptococcus type microbes that can handle the oxygen at the surface, and then more anaerobic microbes hiding out underneath. So each of us have these fascinating gradients of microbes growing on the surfaces of our teeth and hanging out in clusters in our saliva. And this is very normal and healthy, by the way. They're quite unique to each person and each family, and they're affected by health and exposures. And it's actually quite possible to capture the diversity by sequencing the DNA in these types of samples. So in my lab, we've gotten to do a lot of fun projects where we collect sometimes 1,000 saliva samples and sequence all the microbiomes and correlate it back to health status and family and so forth. Okay, so getting back to the beginning, when we're first born, it's actually controversial how much microbial exposure we have, but it's clear that a fetus is exposed to maternal microbiomes, if nothing else, through all the molecules in the blood that were affected by the maternal, like the mom's gut microbiome. But once a baby is born, breastfeeding has a really big impact on the early life gut microbiome. And it turns out that 15% of the energy in breast milk is in the form of special sugars that really we should call fiber, because they're not accessible by human digestion. The only thing that can break down those fibers is a bacteria that carries the right enzymes. So it turns out there are bifidobacterium that can break down the fibers in breast milk. But most of the infants in the US and in the industrialized world are no longer carrying those bifidobacteria. So here's a plot showing you a tour around the world where this kind of turquoise color is representing the type of bifidobacteria that is able to break down breast milk sugars. And you'll see in Bangladesh and Gambia, it's quite abundant, but in almost every other sample in the world, there's very little of that bifidobacteria that can break down the breast milk sugars. This has really big impact on our health. Speaking of increases in allergies and eczema and type 1 diabetes, for example, our immune systems through human history were developing while our guts were dominated by bifido, more like Bangladesh and Gambia shown in this plot. Whereas in the rest of the world, we actually are not having that happen anymore. Having that bifido with a secret nutrient source from the breast milk means that if a pathogen like a salmonella comes through, it can't access that same secret nutrient. And so you're protected from infection. And this is the bacteria that humans had in their gut for such a long time. This is how our immune systems were developing through a lot of human history. So One hypothesis is that lack of access to this bifido is an important part of why allergies and autoimmune diseases are becoming more common. And so actually there's a really interesting set of studies going on right now. For example, there's a study in Europe with 1,000 people where they are giving this bifido to newborn babies and every day for a year. And then they're going to wait, and it's in people who are at high risk of developing type 1 diabetes. And they're going to wait and see if exposure to this bifido protects from the development of type 1 diabetes. So wait a few years, we're going to know a lot more. I'm very interested to see how this goes. 10 years ago, I didn't think we would understand this as well as we do right now. So to me, this is a really exciting progress. Okay, yeah, and here's just more fun stories about the bifido. For example, a group at Davis, they call themselves the Davis Milk Group, discovered these bifidos, and they're being sold at a company called Evolve Biosystems. The product is called Avivo. I don't work for them. I'm just sharing this for fun. And they have found really interesting trends. For example, if you have that bifido and you're breastfed, then a baby's fecal pH, the acidity of the baby poop, will be much lower. And if you look through the last century, you actually see that we had an increase in the baby poop pH, suggesting or correlated with the loss of this bifido. So it's a really interesting moment that we could help to recolonize humanity with this bacteria that we were having in baby guts through our history. But, you know, how do we pick which one to use? There's just really interesting questions around that. And so I've got some resources here if you're interested in that, or feel free to ask more questions. And then just to give an example of here in Orange County, one of my graduate students worked with some doctors at the Children's Hospital of Orange County. We got fecal samples from preterm infants. And just to show you an example, I mean, we were not finding bifido in these samples. We were finding bugs that you really might consider to be more pathogens, you know, Enterococcus and other gram-negatives. Bifido is in dark purple, and you can see it's really not very abundant in any of these samples. So that's a local example. And then when we use this bifido to supplement, you'll see that the bifido does really well. So in part B of this plot, the blue-purple color is bifido. And when the babies are given access to this bifido and they're being breastfed, the bifido comes to dominate quite quickly. And that's more like what was being seen in the Gambia and Bangladesh in the earlier plot. Okay, so I promised you a story about the microbiome through the ages. Here's our baby stories. And anyway, it's been a really interesting 10 years, and I'm very excited and optimistic to hear what happens with the studies with these bifido interventions in the future. So in general, in the lifespan, we expect to see a rapid change in the microbiome from infancy to age 3 as babies and toddlers start to eat. Their gut microbiomes reflect changes to access those nutrients. And then you end up with a more stable and sometimes lower gut microbial diversity in late adulthood. And we know that people eating less diverse diets, for example, sometimes people living in institutions without access to very diverse food, and like a lot of processed food, will have lower gut microbial diversity because access to whole foods can be more difficult in medical and nursing home settings. But it's often possible that physically active older adults who have access to whole foods are able to maintain higher gut microbial diversity, which can even protect from infection in addition to other health states. At a population level, we now have done some really big microbiome studies around the world in the US and Europe, and this is hopefully starting to catch up in all different parts of the world as well. And so some of the things that are the most correlated with our microbiome composition include the use of medication. So antibiotics and other drugs can have a really big impact on our microbiome. And other factors that explain a lot of the information we see in our data include age, BMI or body mass, And like here's fruit consumption showing up as a really important factor, which is interesting. But overall, we actually have a hard time finding clear signals in a lot of the data. It's really, there's a lot to be discovered, I guess is the best way to say this. If a lot of people will do these kind of studies and it's not really quick that we can pick out a correlation between the question you're asking in the study and the kind of microbiome composition that we're detecting. So that leads me to this question, which I think is on a lot of our minds. Should you get a microbiome test? Maybe you've seen ads where you can pay $100 or more to get your microbiome tested. And to be honest, it can be really hard to interpret the data. That was the main message of my last slide is, you know, if you were to do one of these tests, it would be very hard to interpret the data, is the truth. And one of my colleagues in Boston last year took his own sample and sent it to 3 companies and got very different answers back. Now, it's not to say we can't make sense of doing research studies in this context. If we design a study and collect a lot of samples from people and analyze them all the same way and make very careful apple-to-apple comparisons within our study, we can learn something. But it can be harder to learn something as an individual going to one of these companies. So personally, I would not advise trying to get a microbiome test. It's hard to make sense of the data so far. Similarly, or another thing that you might wonder is, say you knew that you had to take antibiotics. Say you knew that you were going to have a surgery or a procedure that required antibiotics, and you want to avoid a negative outcome. Some people end up with chronic infections with Clostridia difficile, for example, after taking an antibiotic. So while this might not be a very palatable suggestion, here I am in this beautiful teaching kitchen, but this is an interesting idea. You could bank your own stool sample. So for example, you could put a sample in the freezer at a moment when you're really healthy, so that if you needed to replenish your own healthy gut microbiome, rather than relying on a donor with unknown risks, you could replenish your own sample. So these are the kind of questions emerging in our field, which has been really fun to think about. I welcome questions on this front. So let's see, it's getting to be close to 12:30. I am going to just back up here for a second and think about what else to share with you before I run out of time here. Okay, so I have a few more slides. I'm going to keep going, and then we'll see how it goes. Oh man, excuse me. Okay, this is a really favorite study showing microbiome data at different ages from people in different parts of the world. So I'm showing you here the microbiome starting at age 2 all the way up to the 80s. And the measure that I'm showing on the y-axis, the tall part of this plot, is essentially gut microbial diversity. So everyone starts out with a lower gut microbial diversity earlier in life. And in blue are the samples from the United States. And you'll see that the people from the United States have their gut microbial diversity plateau at a lower level. After you establish around age 3 to 6, after people have been eating solid food for a while, the microbiome plateaus. But you'll notice that the samples in blue from the United States plateau at a lower number than the samples from India or Malawi, where they tend to have higher gut microbial diversity. And this is maintained through adulthood. So this is really interesting. Why do we have lower microbial diversity in the United States? I mean, that's a really big question related, back to diet and maybe correlated with some of the increased health risks we have for autoimmune diseases and allergies in the United States. This extends even further in this study, where they've got samples from 30 different countries around the world, and you can see that the people from industrialized countries are clustering on the right side of this plot. On this plot, every dot on this plot represents one fecal sample from one person. And the points on the plot that are closer to each other have more similar microbial communities, so more similar microbiomes. And you'll see the industrialized points on the plot are all clustered together on the right side, whereas the samples from the non-industrialized parts of the world where they're eating more plant fiber and exposed to less antibiotics and so forth tend to cluster together as well. And we can see some really interesting differences in microbial community composition. Many of these bugs are very familiar to me. And for example, Prevotella is famous for carrying genes that can break down plant fibers. So this to me is an indicator of somebody who's eating a lot of plants. And you see that a lot more in the non-industrialized parts of the world. Whereas the samples over here from the US and the other industrialized parts of the world are rich in bugs that can break down mucus. These verruco microbes are famous for breaking down gut mucus, indicating that the people in this study were not eating very much fiber, and the bugs in their gut had to turn to eating the mucus lining the gut because they had no fiber to eat. So, the message here is eating fiber is really important for maintaining a healthy gut microbiome, and that's going to be a theme in our lecture series this year. We're gonna talk about that a lot more. But I do want to hand it over to Chef Jess, and I will hammer home the idea about eating more fiber in future lectures. And here it is with the recipes. I will share once more so that you guys can find the link. We've got spicy beet salad, multi-seed crackers, and dark chocolate berry bark. And I want to thank my lab. I have a really fun group of students and postdocs working with me here. And we've got funding from a number of agencies making our work possible. And I have a final set of take-home messages that I want to make sure you get to see today. And I think we'll be sending these around as well. Just that microbiomes are unique to each person. Most microbes are not pathogens. The early life gut microbiome is very influenced by breastfeeding. Anyhow, these are my big take-homes from our first microbiome lecture. And I will actually answer questions, but I'm going to hand it over to Chef Jess.
Okay, so we are going to be making 3 high fiber dishes. And I'm going to start with the bark of all things, because I think it's fun to start with dessert, maybe. So we're working with chocolate. And so for this dark chocolate berry bark, a lot of people give chocolate a bad rap sometimes because they think that it's all sugar. Guess what? It has fiber inside of it too. So chocolate is one of those cool things that you can work with, but you need to buy the right chocolate. Most milk chocolate has a lot more sugar in it than anything else. So I like to buy dark chocolate. So the darker, the better. Trader Joe's does a good job of like their pound plus. But I think the most important thing to remind people about chocolate is that it's sensitive. It does not like water. It hates water. So as long as you get water inside of there, it's going to seize up. You can melt your chocolate over a double boiler, or you can do it in your microwave, 30-second intervals. So you just do 30 seconds at a time and then just let it melt on its own. Don't force it to melt. Let it sit and kind of do its own thing. Now over here, I have white chocolate. And white chocolate is even funkier than dark chocolate is. It melts at a lower temperature, and so you definitely don't want to overdo white chocolate. But if you notice, I have a little bit of fat in all of them. So we're using coconut oil. You can use anything that you have on hand, but the fat helps keep the chocolate together from seizing. So what we're going to add in here are nuts and berries. So nuts are a great addition. But you do need to remember that there is fat in nuts. So you just want to just be careful. Good fat though. And so we've got some walnuts here. You can toast them up if you want to. And again, you can use your microwave to toast it. But what I'm going to do is I'm going to add the nuts into the chocolate. I'm going to give it a good mix. And then I'm going to chop up my berries too. And it's quite a lot of berries. It's a lot more berries than fruit for the reason of you want this to be mainly fruit and not much chocolate. Not that there's anything wrong. What?
We're getting asked if you can put the camera.
Oh, whoops. I am.
Great. Yeah,
I am
good.
so
So we got
sorry.
a great question.
I got, yep. Sorry, everyone.
So,
So it was like this. So just so that you can see, I apologize.
I should have noticed that too.
No, it's like one of those things you don't realize until you start. So
Well, I can see.
yeah, I know. I was like, oh no. And we had it all down.
It looks really good.
Yeah.
Okay, hopefully you guys can all see. Questions welcome. And actually, we got a question I wanted to answer
Go
about
for it.
what can people do to assist their gut microbiomes?
Mm.
And really important thing you can do is eating well. And so eating well includes getting a diversity of different kinds of plants into your diet, and especially fiber. I think there's so much focus in our media about eating enough protein, but actually eating fiber, which are just complex carbohydrates that persist in your gut and make it all the way down to your colon where your microbes are the most dense. You know, most of your microbes are down in your colon. So you have to eat fibers, lentils, beans, chia, you heard Chef Jess saying chocolate's high in fiber. All those things will help you. That's probably the best and most important tip for improving your gut microbiome.
So something like this, like if you have a sweet tooth and you need something sweet, this is great because not only are you getting the fiber from the chocolate, you're also getting it from the berries and the nuts that are inside. So you can use what you want. So I'm just going to spread this out. And then you do want to keep this in your freezer. The main reason being that you're adding so much berries to this that it'll not break off, but it's not going to solidify very well unless it's in the freezer. So I'm going to stick this in the freezer and then you'll break it apart like you would bark. Now, what does that do if I keep it in the freezer though? It can't be that shiny, shiny chocolate. So the thing with chocolate is that it doesn't like rough temperatures, or it doesn't like random, not room temperatures. And so it blooms. That's when you see it get white on you and things like that. And it's just a temperature shock. There's nothing that changes the flavor of it. So you don't need to worry about it. But if you want, you can then also, the white chocolate is just really excess. You don't really need to add it if you don't want. It's just pretty to have that variety of color. But then you can put this in your freezer and then just cut it into pieces, break it off once it's solid, and use it as a snack. And then just keep it in your freezer or refrigerator, I guess, after it's frozen and you've got it ready to go. And it's a super easy way. And if you don't like berries, you could use apples. Any kind of fruit that you want, you could probably get in there. And I'll be honest, you could probably sneak in some spinach or like some kind of green in there and put it away. Can you guys put this in the freezer for me? Thank you. All right. So for the next one, we're going to do a beet salad. Do you like beets?
I love beets.
You love beets. See, I hate beets. And so when I was looking up stuff, because beets are incredibly good for you. And so it's like, okay, how do you make beets edible for people who don't like beets like me? Because the thing about beets is they're very earthy. They're like, they taste like dirt to me.
Even fresh.
Even fresh.
We chop fresh red and yellow beets and into small pieces in our salad, or
grating it would probably be good. No, I just still can taste it. I will say the yellow beets do tend to be a little bit lighter than the red ones. But what I find funny is that beets come in all different sizes. Look at how small these guys are today. So these are tiny, tiny little beets. They're organic though. And so if they're this small, don't peel them. You actually don't need to peel beets at all. Because remember, the skin is where a lot of the fiber and the nutrients are, especially if you're buying them organic. So some of the things you can do is you can just chop these up. So I'm just going to take off the top, and they're so pretty inside, I know. And we're going to actually roast these with the skin on. So I'm just quartering them. So you can cut them into small bite-sized pieces if you want, or I'm just going to quarter them. and keep them whole just like that. Now, if you buy organic beets, you usually get left with this whole thing. You can cook all of this. So just a little bit of garlic, oil, olive oil, and you've got like sauteed beet greens, or you can make pesto out of it as well. So you can take greens and make pesto out of them. Okay, so I've got those ready to go. I'm going to add a little bit of oil. And this is what I've done to make them edible for myself. A ton of spices. So cumin, ancho chili pepper, a little smoked paprika, and a pinch of cayenne. And you're going to just really toss them in here. And the recipe calls for 2 pounds, and this is not 2 pounds. And so there's a lot more seasoning on here than needs to be. But then you would take them and you can put them on a cookie sheet without all the extra spices, take those off. And then they roast up into these tiny little kind of coated, like majorly coated pieces of sweet potato, beets, right? Like that. So then we're going to build the salad. And again, to keep it spicy, we're going to use arugula. So I've got a little bit of arugula and spinach. So if you've never had arugula before, arugula has a tendency to be a little bit spicy. So it's nice to have for something like this if you're not used to having beets and things like that. The other ingredient that goes in here that I don't have is goat cheese. I have regular Parmesan cheese, but you can use any kind of cheese that you have on hand. And then,
Yeah, we got a question. We got a couple questions
Go
coming
for
in
it.
too. So one of them is about psyllium husks.
Go for
which, psyllium husks are plant fiber. So it's not, it's, you buy it in a jar and in a way it feels like a processed food, but at least it's coming from a plant. So if I had to choose between Miralax or psyllium husks, which is Metamucil, I would definitely go for the Metamucil for the psyllium husk because it's from plant fiber. But your best bet, if you can, is to be eating dietary fiber, like getting from different plants.
It is a little bit, it can get hard, I understand. And so the chia seeds?
Right.
Yeah.
So another kind of intermediate suggestion is chia seeds because they have huge amounts of fiber.
Yeah.
So a serving of chia seeds, 2.5 tablespoons, has about 10 grams of fiber in it. And most of us are getting half of the daily recommended amount, which is about 30 grams a day. And so if you're getting half of that and you boost it up with a serving of chia seeds, that will really help you get there,
I think, it's just remembering. And if you're a smoothie drinker, and so one of the big things, like a lot of smoothie drinkers, they can add a lot of fiber in, but then it's funny because one of the things that most smoothie drinkers add are protein powders. They're constantly adding like protein powders. And so it's like just as much protein powder you're adding in, you can add just as much fiber into your smoothies.
Yeah.
And if you're worried about it, this is the trick. I was like, just add hemp seeds into everything. I was like, because the hemp seed has the protein, not as much as I understand it's not, but it's a pretty high protein. And then you've also got the fiber from it. So it's one of those like kind of cheats that's more, it's not a processed powder that you don't know, because protein powders can get pretty expensive too, the good ones. And it's like protein powder, as I was talking to our registered dietitian, it's one of those things that, you have to pay a pretty penny for if you want the good stuff. And if you're not getting good protein, then you really shouldn't be wasting your time.
The protein powders can be, you don't know what's in them. Really.
Exactly.
Processed food.
Yep.
So, I mean, even chia seeds actually have quite a bit of protein in them.
Yep. So they could work.
Hemp. If you go to that section of Trader Joe's that has the chia seeds, and flax seeds, and the hemp seeds, those are all things you could add to your smoothies and get both fiber and protein.
And on Monday, sorry, we're going to work on the salad in a sec. On Monday, though, see, this is what happens when we get to be next to each other. We did a smoothie, and I had upset patients at me. I had somebody actually walk out of my cooking class for once in my life. And the reason was, and I understand, I was trying to teach people how to make smoothies. And the smoothie recipe was black beans, maple syrup, cocoa powder. Oh no, not maple syrup, dates, and then cauliflower, like frozen cauliflower. And he looked at me and he's like, yeah, no, I need to leave right now. And so I was like, yeah, it's okay, because it's like one of those things. And let me just say, there are people who made it and loved it. Like they ate it like ice cream. Me and my students made it and I could taste each piece of cauliflower in there. And so it's like one of those things. You just have to try it though. And it's like, it's these cool ways that we can get fiber into our diet. And it's like, what else can we do? And black beans in a smoothie actually work. So the black bean part was okay for me. For them, that wasn't okay. But you could add black beans into a lot of things and it goes pretty smoothly. And there's your fiber. So
For sure.
all right, so let's talk about a really simple dressing. So we just have balsamic vinegar, a little bit of honey or some kind of sweetener, and then mustard. So mustard is our binder. So this is what's going to hold everything together. If you have mustard around, it's fine. You don't need to use it, but it is what keeps it together. It's the emulsifier. And then I have olive oil, and I'm just going to drizzle in olive oil. Now, because this dressing has absolutely nothing but olive oil, vinegar, and a little bit of the mustard, this dressing will actually stay in my refrigerator for up to 3 weeks. As soon as I start adding anything fresh in there, like fresh garlic or fresh herbs, it'll only last about 3 to 5 days. So I'm not going to dress this now, but I want you to know, if you want to taste this, don't use your finger. Don't use a spoon. Use what you're going to eat it on. So say you are eating it with salad. Dip the lettuce leaf in there and then taste it. And then you'll get the exact right amount of seasoning. So that's for that one. And then the last one is super fun. But you will have to play around with it a little bit, because I'm going to show you, because that's why.
So these are the multi-seed crackers?
These are the multi-seed crackers. So I had my students make this for me earlier this morning because it needs to sit for a while. I realized why mine didn't turn out exactly the way that it needs to, and I'll show you in a sec. So you're going to use raw. So first of all, it's important to make sure everything you use is raw. You don't want anything toasted. We're going to cook this at a very, very low temperature, and because of that, it'll toast up. It'll actually get brown on you. So if you're using something that's already toasted, it's just going to burn. So I've got sunflower seeds and pumpkin seeds in here, and then sesame seeds, raw sesame seeds, okay? And chia seeds. We've got chia seeds and not flaxseed meal. We want to use flax seeds, okay? They made the mistake and used flaxseed meal, and that doesn't work as well. You actually want the flax seed because it'll bind a little bit better. This doesn't bind as well because only the parts where the flax seed meal is will it actually bake up. When you soak the flax seeds whole, they gel up even more than this. And so this didn't gel up as much as I wanted. But I'm going to add water. So half a cup of water. Okay. And then we're just going to let that sit for 30 minutes. After you let it sit for 30 minutes, it'll get kind of gooey and gummy and look kind of funky on you. You're going to spread it on a baking sheet. And then you're going to bake it in an oven at 285 degrees. Oops. And it's going to turn golden brown, just like this. Okay. So you've got this nice sheet of cracker. Now you can go in and actually just break it off now. So you can break off the pieces, and you should get pretty large pieces off of it. Now, I didn't add any seasoning into this, but you can play around. So say you want something salty, you could add garlic and something else. But play around with it. If you want something sweet, you could do cinnamon and cloves, cardamom. So play around with it, make what you want, and you could even make this into granola. So if you think about it, it's that same idea of baking all of these whole And then you've got these crackers ready to eat. Now, everything in here is super high fiber and in its whole form too. So you're getting the pumpkin seeds whole, the sesame seeds whole, and the flax seeds whole too, if you were to add the flax seeds. But the whole idea is that you want this mixture to be very gummy. So you can see it's already starting to soak it up and get there. But you want to be able to do as thin of a layer as possible so that it's nice and crispy, so you can crisp it up and then add it in. So that's it for the multi-seed cracker. And you can keep this in your freezer. You can even put it in your pantry for quite a while. I realized for my salad, I forgot to add 2 things, so I'm going to add them in right now so that I can at least show you some knife skills before I let you go.
Oh, knife skills.
Yep. So just a little bit more knife skills before I let you go. Because the salad is spicy. The spices are spice. So the best addition to something like this is like an apple for the crunch and the sweetness. So I'm just going to cut the side of it. Now it's rolling around, so it's not safe. What do? I don't want to do this. So what I'm actually going to do is I'm going to give myself another angle. I'm going to cut that in half and then roll it on its side and then make slices out of this because I want matchsticks. Now the apple, if you peel the skin, you lose 25% of the fiber content. 25%.
Wow.
So think about that the next time you buy apples. Buy them organic because they are part of the Dirty Dozen. They are one of the few that we want to definitely buy organic because if you eat the skin, that's the part where the pesticides hit and things like that. And so the skin is where it's at. I'm going to add more in just like that. And then a red onion too, or part of a red onion, because I know we have beets in there. We always encourage people to eat the colors of the rainbow. And the one color people struggle with is purple or red, purple or blue. And your red onion gets you there. So I cut it off. I keep this intact, the root end. So it grows up like that. I'm going to take off that frilly part and then follow the lines just like that. Slice in once. And then I've got a dice. Now, if you feel like dicing onions are frustrating, dice more and keep it in your refrigerator. Because it's one of the things that builds a lot of flavor and makes and breaks a lot of dishes. So if you want to start to cook more, one of the things you can do is slice or cut more onions. And there we go. And there you've got a little bit of cheese in here would be good. Goat cheese. And then you've got a spicy beet salad ready to go. All right. And that is it. If you have any more questions, we'll be here hanging out. Let us know. Well, that's all I have.
Great. Well, we did get one more question about how much fermented food to eat and ideas for fermented foods. And I did just type the answer, but I would say we don't know the answer to exactly how much fermented food each person needs. That's a hard question to answer. But for example, the Phi-FO study at Stanford, they had yogurt at breakfast, kombucha at lunch, and kimchi at dinner. And that's, maybe that's overkill, but finding a way to get fermented food into every day is a good idea. And some of those are just really easy that people like. Or you can use, this salad dressing, for example, has balsamic vinegar. Another trick is miso or using apple cider vinegar that still has the mother in there.
Yes. So Fermentation Farm is a great resource if you're interested. Feel like a lot of their products are good, but it is pretty easy to make your own. And we did a lacto fermentation class. And so if you go back, you can look up some recipes for lacto fermentation and try them at home. I think that there's some really good ones that you can try to do on your own, and making them on your own seems to be a little bit better too, because a lot of the times, like say kimchi's a great example. Kimchi's high in sodium. So you want to be careful with a lot of fermented foods because they tend to be high in sodium. Or if you look at kombucha, it might be high in sugars, but it's different sugars. And so you have to do the research behind it too, because you're not ingesting all those sugars from the kombucha. It's been eaten by the mother, but they had to label it a certain way.
Yeah.
And so I feel like with fermented foods, labeling gets kind of weird too, because you add so many things into it, but there's like process going on with everything that's eating it away too. So it's not necessarily bad for you, but I think with like everything we eat, it's all personal too. Like we need to figure out like and some people might be able to eat kimchi and some people might not be able to have miso. It really just depends. And so play around with it and see. And so this is our bark. They just brought it out for me to show you, but it's not there yet, I don't think. Almost there, but you should be able to like almost snap it off. So, but that's what it would look like. And then you would be able to have some nice berry bark ready to go too. Thank you.
We got another question about the nutrition values of the recipes.
Ooh, I can analyze them for you. I will get them. I'm not a proponent. I do not supply to this company, but one of my favorite ones right now is called Happy Forks. And the reason why I love Happy Forks is because it does a omega-3 to omega-6 balance. And I think that in our American diet is very, very high in omega-6. So we need to be careful. Not that there's anything wrong with omega-6s, but there's a balance that we're trying to get with 3 and 6. And so it's how do we get more of that balance in our diet? And that website, happyforks.com, you can actually just put in the whole recipe and they'll analyze it for you. And they use numbers from the USDA. The only problem that I'm having with them is that a lot of our new superfoods like hemp seeds, sorry for using the word superfoods because I don't like using it, but like things like hemp seeds or like flax seeds aren't necessarily on there to be analyzed. And so you aren't getting exact proportions yet, but it's really cool with the comparisons of 3s and 6s for the omegas.
That's great. So Chef Jess, sounds like you're willing to add that to
I will add that. Yes, I will add that on there as well.
Very cool. All right. Sounds great. Well, thank you.
Thank you, everyone.
Thank you very much. Happy Friday.
Happy Friday 13th too. Yes. Good luck out there. No.